Alright, so I'm going to give you a chance to take a picture of this with your phone, because this picture you need to know, without a doubt. So, it's also in your book in a different form, but if you don't have a book, or take a picture of that with your expensive thing that you're using at Minutes On, whatever. I'll give you a few minutes to pretend that's Brad Pitt, you're taking his picture. While you're doing that, I'm going to go up and start writing some stuff on the board that we're going to use this chart to do.
Do you have this on your website?
It is, on the thumbnails. Yes.
But it's not
Also online. Yeah. I know if it isn't on your phone, it's not real. So, when I was in college, we didn't have them phones, right?
Is that okay?
Everyone have it? Is there satisfaction? You're supposed to be. I'm just going to illustrate it a different way. Oh, sorry. Okay, so what I'm going to do is I'm going to take that picture, which you're going to have to just learn, and write it out in a table form. So what we're going to do is we're going to figure out where in the nephron we're talking about, the chemicals that are getting dealt with, and how we're dealing with them. So we're going to start at my PCT. From lab, what did that stand for again? Proximal convoluted tibial. On this diagram, that is right here, this white area here. So the PCT is this box. So now we're going to fill in the chemicals that your nephron is trying to process. Someone name a chemical for me off that chart. No, you're looking in the white box, you're naming these. Try that. Someone say something. All right. Tough crowd here tonight. What else? Bicarbonate, HCO3 minus, and
glucose and amino
there's
acids.
glucose. There's amino acids. There's also, what else? There's H2O. And there's some poison, which is always ambiguous, and some H+. So here's a listing of chemicals that my nephron is going to deal with in my PCT. What we're going to do now under the green color is what my nephron is going to do with those chemicals. So you're looking at the arrows and the direction the arrows are on the picture. So let's see if you can figure this out. Looking at, let's pick one. Let's look at glucose. Relax. Where's the arrow going in this picture for glucose? From where to where? Let's find it. Here's the arrow. It's going from here to there. So that means from my what to my where? From the PCT or the nephron back into my body, right? So what did we call that last week? You can go from the nephron back to the body. Reabsorption. Reabsorption. So my glucose and AAs are going to be reabsorbed. Remember that from last week? We're going from the nephron, which is in yellow, back into the body, which is off the screen. So I'm reabsorbing my glucose and AAs. Look at the other ones. Tell me about the sodium and the water. What are they doing? Being reabsorbed? Is the arrow the same direction? Let's find out. Yes, they're all the same. Therefore, those are also reabsorbed. They're going from my nephron back into my body. Look at the H+ and the poison. Look at the arrow there. What do you see? What was it? I heard it. Secretion. How'd you know it's secretion?
Body into the nephron.
That's right. It's going from outside my nephron body into the tube. Remember, that was secretion. After I filter, I'm adding to the nephron. So we're going to put that as secretion. So I'm reabsorbing these water, salt, and bicarb. I'm reabsorbing glucose and AAs. And I'm secreting or getting rid of the H+ and poison. Let's review why. Why would you reabsorb all this stuff up here? I can use it, right? Sugar's nice. Amino acids are great. Those are useful. Why am I secreting these? I don't want poison, right? In theory. I don't want acid. So it makes sense that my body's going to reabsorb whatever came in my filtrate. I'm going to reabsorb. And I can also add stuff back. Make sense? See the logic of the picture? I have to add one more detail PCC says thou shalt know, which is the color of the arrow, which is around here. So let's look at sodium chloride. How is it reabsorbed? What kind of motion transport is it using? Active transport. If you look here, that color means active sodium chloride. So I'm going to circle it. I'm going to say active. The way I'm going to do this is I'm only going to notice the active ones. You just assume the other ones are not active. Can someone find another active one for me on this picture? Glucose and AAs. Those are also done through an active process. Remind me from Biology 112 what active meant.
You need
You need to use energy or, ATP. So the ones I didn't circle up here that are being reabsorbed, how are they being reabsorbed if I didn't circle them? Passive, which is the main diffusion. Look at the two I'm going to secrete. What kind of process is secretion? Active. Because if I need to secrete it, I have to pump it. Therefore, there's only active transport in secretion. I have to use energy to get rid of it. So at my PCT, I'm going to be using these chemicals to do this job this way. That is the answer you're going to give me on your next exam. It's exactly how I wrote it here, more or less, in words. What, where, how, and what it's doing. Make sense? Let's keep going. What's the next part? The descending loop. Loop of Henle, which you learned in lab was this long tube coming down. Tell me, what chemical am I doing on my descending loop?
Water.
Water. Very good. What am I doing to the water on my descending loop? I have a vote for reabsorbing. I have a nod. I have confusion. Let's see, the arrow is going from nephron out, which means reabsorb. So again, I'm going to be reabsorbing water. Why? I like water. It's kind of nice. What process am I using to reabsorb the water? Passive. So I'm not going to circle it in the way I'm doing it. That's a passive or osmosis or diffusion. Let's now do our ascending loop. The way up, that's this loop. Over here, from lab you learned is this one. What am I transporting in my ascending loop? Sodium chloride. I'm going to list it twice, and you'll see there's a reason why. What am I doing to the sodium chloride? What process am I doing to it? Reabsorption. Again, I'm leaving my tubule and heading into my body. So I'm going to reabsorb. I like salt. How is my sodium being reabsorbed? Active or passive? Both. That's why I listed it twice. So there's an active component, which is higher up. There's a passive component, which is further down. But I'm using both processes for sodium transport in the ascending tube. So there's a passive and an active. So far, so good? Okay. We're going to go over here now. Do my last couple parts. My DCT, my distal convolution, goes to the white box over there. Make the same list. What chemicals am I moving into DCT? You can read them off the chart, right? Let's do it in. NaCl, HCO3 minus. We always have H plus. We have this nebulous poison which no one can identify. All right. Let's figure out what they do. So what am I doing to the NaCl and the HCO3? Are you sure?
Yeah.
Yes, you are. So reabsorption. I'm leaving my nephron and going into my body. I'm going to reabsorb. What am I doing to the H plus and the poisons? I hope I'm secreting them, otherwise I'm dead. So I'm putting them into my urine. So secretion. Let's do the easy one. For the secretion, what kind of process are you going to use? There's only one way. It's active. I'm going to circle those and put active. I always have to use active energy, active transport for secretion. But the other two, NaCl and HCO3, those are also active. That's okay. Perfectly allowed. So I'm going to use active process here as well. That leaves the last part, which is right here, which is my collecting duct. tell me, what am I doing in my collecting duct? What chemicals am I doing in my collecting duct? Water, NaCl, and urea. We'll talk about a little later tonight. What am I doing with the water, the NaCl, and the urea? Are you sure? Yes. Hope so. So I'm going to reabsorb those. So again, reabsorption, I'm leaving the nephron and going into my body. Let's go through the processes. The water, how is it moving? It's passive, so I'm not going to circle it. The sodium chloride is moving how? Active, so I'm going to circle that one. The urea, also passive water. So what you're supposed to do, PCC says, is memorize basically what I put on the board. The sections of the nephron, the chemicals that move, why they're moving, and how you get them to move in the first place. Right? So thou shalt do that, thou shalt. So what I'm going to do now is show you animations about this process. So give me just a second to bring it on the screen. It'll take me just a second to bring it up. Go through that.
Why are we reabsorbing urea?
That's a good question. Give me about half an hour, I'll make it make sense. You do it to do osmosis quickly. Give me a second to put it on the screen. Where I put it? There we go. It's actually made for high school students. Don't be, offended that I'm giving a high school animation here. But this is actually the best way to learn. So here's my kidney. Let's zoom on in. Zooming in far. All right. So what's that thing there? That's supposed to represent the nephron. So if we here's the tubing, there's the glomerulus we talked about last week, there's some blood vessels collecting a lot of stuff we kind of learned in lab. Right? What we're going to do is we're basically going to zoom in a little more, and we're going to look at it like this. So we again have our blood going in, that's the corpuscle you learned in lab, that's the glomerulus, whatever you want to call it. PCT, down loop, up loop, DCT, collecting duct. The same things are on the board. Right? I love this animation because I can pick chemicals and see what they're doing. Let's pick one. Glucose is fun. So, if I hit start, what do you think the glucose will do? Walk me through it. Where do you think it'll go? Will it go here? Yes, because glucose is small. It should enter my kidney, enter my nephron. Then what will it do? According to the thing you just wrote on the board, what should it do? Where should it be reabsorbed? Let's see if that's true. Now I'm in the nephron. Ooh, let's do that again. Bioporn. Let's watch that again. Let's watch the glucose. You watch right here. Bingo. It went from my PCT, or my tubule, was reabsorbed into my bloodstream. So now it's no longer in the P, it's now basically back in me. It's a silly rhyme. That works. Why do I want to reabsorb glucose? I like it as a rule, right? It's energy. And so I'm going to then send it back to my body. So that the PCT is going to have the ability to reabsorb the glucose. Right, let's pick another one. Let's do protein. You're going to see. Why didn't it do anything? Right, that's that too big idea from last week. So if it's big like a protein, shouldn't do anything, just stay in my blood. Right? Alright. Let's do another one. Let's do sodium. So when you do sodium, let's watch sodium. It'll be a little blue thing. Fast little guy.
Oh.
Whoa.
He cloned
Whoa.
himself.
Yeah,
Can't trust that.
this is not anatomically correct, right? You get that? Let's play that one more time and then try to explain what you saw. Okay, so I'm getting this part. There's my sodium, it's small enough to go in. What did that just illustrate right there? Reabsorption. So I reabsorb some in PCT. So keep watching. Oh, do it again. So reabsorb. Whoa, what's that? Showing reabsorption again. Remember, it happens in more than one place. So the animation just shows it as the ball clones itself, which it doesn't. But where is this? What part of my body is this one where I reabsorb sodium? The ascending loop. So I reabsorb some in my PCT. I can reabsorb some in my ascending loop. Look over here, I can do some in my DCT. So there's more than one place you can reabsorb it, and that is what the animation is trying to show by having the blue ball split. Right, insert joke now. Right? Let's do another one. Let's do water. Here we go. What do you think you'll see?
To go all the way.
There's one. There's two. All right. Should be one more. So why are there 3 little water balls bouncing around?
Because we love water.
Yes, same thing. I'm reabsorbing it in multiple places. If you watched the animation, you would have noticed there's some PCT, the down loop over here. What this animation is trying to show you is that different chemicals are absorbed different places, some more than once. But every chemical has a spot on your nephron where you would absorb it or secrete it. Right? That's why you're going to memorize that picture in this table. Make sense? So what we're going to do now is go through the biology of how the chemicals actually do that. I'm going to show you a different animation for a second. Step it down for a minute. All right, this is actually Biology 112 again, that place you hate so much. Okay, it's going to zoom in. I'm going to actually replay it so you can see where it zoomed one more time. So if you look, I wish there was a way to pause it, but you just can't do pause there. So on these animations, the way they're written in program, is where it says tubular lumen, in English, that's the tubule, that's the nephron, that's urine over there on the left. Anyone see when we started what was on the right? What's that over there? That's blood, right? That's a capillary. So in English, the P's on the left, I'm on the right. Right? This is the border between the nephron tubule and the rest of my body. So notice the phospholipid bilayer? Does that ring a bell? All right, so if I replay, urine's on the left, blood's on the right, P and me. And if you remember Biology 112, you know what these things are. What are those things in the membrane?
Protein carriers.
Protein channels, transmembrane proteins, receptors, enzyme channels, 231, you have gated, all that stuff. Remember all that stuff we've been talking about, right? Use any of those words, I don't care. But here's some kind of molecule that transports channels, receptors, pick a word. If you remember Biology 112, enzymes are specific, right? So we're going to click one. I'm going to click a sodium channel. What should go through a sodium channel?
Sodium.
I hope so, otherwise I've been lied to all these years. Right? So now we have to figure out what the sodium's doing when it goes through the channel. So looking at the direction of the little balls, what's sodium doing? I vote for reabsorption. I have a hand raised. I got amens. Yes. How do you know that's reabsorption? It's going from where to where? From P to me. Right? P's over here, B's over there. So I'm reabsorbing sodium through a channel. Right? Let's do another one. So what do you see there? Yeah, sodium glucose co-transport. So I'm reabsorbing sodium and sugar at the same time. I'm still reabsorbing them.
Does the sodium help reduce energy costs to do
No,
that?
I've never heard that. It may. I've never heard anyone make that statement because I've never read up that much on it. I think it's just the way the carrier molecule is. Let's try this one. Tell me what that one's doing.
Hydrogen. Acid's out, sodium in.
Very good. So acid is out, H plus is out, sodium's in. So therefore the sodium I'm reabsorbing, but the acid I'm doing what to?
Secreting.
Secreting. In fact, that's what you wrote up there, right? H plus is always secreted. So this enzyme is pumping something out of my body into my urine, acid, because I don't want it, and pumping sodium from my urine back in because I do want it. So the take-home message you want to kind of deal with in your head is everything you learned in Biology 112 is on the board. Every section of your nephron that does something is because it has an enzyme that does it. Right? So, if over here, if I am secreting H plus in my DCT, but I'm not secreting H plus in my loop, why am I secreting it there and not over here? The answer is, I don't have the enzyme here. I have it over there. So everything you drew on the board is because the enzymes or channels are in those locations. You have glucose channels here, but not here. You have water channels here, there, and there. So everything you learned in 112 about enzymes is coming back to haunt you. So the enzymes are what determine this process. What goes where and how.
Except some things that are going passively, like if
Yes.
it's just like an ion or something, have they, or not an ion, but like a
In this case, actually,
water molecules?
they still have to have a channel.
They do?
Yes.
Okay,
The hydrofluoric to work. You're right. They could do passively, but most of it is through a channel anyway.
okay.
Yes.
When people get kidney stones,
Yes.
is something in here broken? Is this what
It can
the
be.
broken part is?
It can be a calcium channel. It's not like the calcium out, so you just build up a rock. It's like a pearl, basically. Right?
But it can't be, meaning there are other things.
Yes, it can just be genetics, know how you do it. It can be calcium diet. It can just be GWIZ.
But this filtration process is where that would take
Yes.
place.
Somewhere between your tubes and toilet bowl. You understand this concept of the enzymes? So when you're looking at this board that you have to memorize, you think about every one of those things in the direction and how is based upon these little enzymes doing their job. So if you remember 112, which none of you do, right? Then you should be able to figure this one out. Suppose I ate a lot of chocolate cake, or whatever, and I have lots and lots of sugar just floating in my pee. First off, why is the sugar in my pee at all? Because I can be filtered, right? It's small. It ends up in my nephron. Then I have these little enzymes whose job it is to reabsorb my sugar. How much sugar will they reabsorb if I have a ton of it? As much as it can. But how much is that? Right to right. Remember your Biology 112 experiment with that saturation curve with the substrates? You vaguely remember that? If you add more substrate, it eventually flatlines. K-max? No one cares. No one learned anything that class. Okay. No. So let's look at our choices. Which of these four things would occur if I have a ton of sugar just floating in my urine? Tell me which one it would be.
Just one?
There's only one. Will the enzymes work faster? No, you really can't control their speed, right? That doesn't happen. Will some of the sugar not be reabsorbed? I can buy that. Will it take longer? Not miss sense. And will you magically make more enzymes?
No.
No. The best choice is?
Not all.
Right. So you're overwhelming or overloading or oversaturating the enzymes. They only have so many enzymes to move so much sugar. So if I have more sugar than they can work on, they just don't work on. Right? So let's do an analogy. They do not live in your kidney. There are people on my TV, but those are not them. Right? They represent the channels. Yes?
Sorry.
Yes.
They don't absorb the extra glucose you just ingested. So
Yes.
then what does it do?
That's what we're going to do here.
Oh,
Watch these people. They represent the enzymes of the channels. This is my nephron. The toilet bowl's off the screen. Here we go. What are they doing to the sugar in my urine? They're reabsorbing. They're taking it out of the urine stream, out of the tubule, out of the nephron, out of the filtrate, pick a word. And they're going to put it back into my body. They're reabsorbing sugar because it's a useful thing to have. Make sense? So they represent the enzymes that are doing over there. Let me know when you're bored watching them. I'll do this for an hour, right? I think it's fantastic. More sugar.
More sugar.
More sugar? All right. Here we go. Now I've eaten lots of candy bars. Let's watch them. Must be a union job. So what do you notice happening? Right. The sugar is higher than the enzymes can process, or what they call Vmax transport. So your doctor would say you're spilling over sugar, and that's actually the analogy they would use. The sugar just keeps coming on out because I'm no longer reabsorbing all of it. I've oversaturated my enzymes, so I'm spilling over. So give me a disease that you know where you pee sugar.
Diabetes.
Welcome to diabetes, right? In diabetes, you have so much sugar because you can't use it. Your kidneys end up spilling it over because you can't reabsorb it all. So you pee sugar because of this. You overwhelm the enzymes. Another way to think about it is you don't want more sugar. You have too much anyway. You just let it go. So the tubules would simply let the sugar keep going. Make sense? So if you're diabetic at the nursing home, they're checking your pee. That's what they're looking at, is not the people. That's the idea.
And also the ketones.
Yes, and the ketones would just prove that you're not using the sugar that you're spilling out. Make sense? Enzyme kinetics. If you understood that concept, then I'm going to show you this picture. This again is a macro view. P's on the left, B's on the right. So there's my blood, there's my filtrate. Notice things are going through the tubule from left to right. That'd be reabsorption. Some things, though, go from the right to the left. That'd be secretion. Hey, what's that little magic flash? I just saw that. That's kind of cool.
Energy?
That's energy. So some of the processes are active, some are not. But you get the idea that everywhere along here you'd see enzymes, or channels, or tubules, or whatever they're called, that are going to be allowing those chemicals to go back and forth. Okay. Make sense? All right. So in our descending loop, it's the same kind of math. Right? In my descending loop, I allow water to be reabsorbed through the loop wall.